Hi, my name is Giannakoulias, and I'm going to introduce you to our study biomolecular simulation-based machine learning models that accurately predict sites of tolerability to the unnatural amino acid acridonylalanine. The motivation for this study was to develop a computational tool that could be used to assess whether or not residues in proteins can be safely mutated to unnatural amino acids without huge deviations in their protein solubility and yield. The reason for this is that there are countless numbers of applications of unnatural amino acids in the world of chemical biology, but it's well understood that identification of which site to incorporate the UAA at is a principal challenge. The method that we developed in our lab in order to tackle this problem consists of 3 steps. The first being biomolecular simulation of the mutation to the unnatural amino acid. In our case, we use the Rosetta modeling suite. The second step being feature extraction, in which we perform an energetic analysis according to the score function used for simulation from Rosetta. And then third step being machine learning for, in this case, a classification task of determining whether or not a residue will not be tolerated or will be tolerated. Here in the bottom left, I'm showing the Lexate protein, one of the 2 proteins that we investigated in this study, where the experimental data shows that the positions in red are not well tolerated by mutation to acridone, whereas the positions in green are well tolerated to mutation to acridone. In the bottom middle here, I'm showing a set of confusion matrices, which are which allow us to assess the quality of our machine learning models. In the first column, I'm showing what's called a dummy classifier, and that gives you a sort of understanding of the accuracy that you would achieve from just random guessing according to the distributions of your data. The way to read a confusion matrix is the x-axis here is the true class, meaning that anything that's a 1 in reality would correspond to like a green position over here. It was well tolerated. Anything that's a 0 would correspond to a red position. The y-axis here is the predicted class. So this is how well this is how the model actually predicted the position. So as you can see, where you'd have coordinates 1, 1, this means that the model predicted the position to be active, where in reality it was active. And so these are your true positives. In the bottom right, you'd have the coordinate 0, 0, which are where the model predicted the position to be inactive, and it was inactive. So you'd like to maximize the percentages in these 2 categories. Up here, you have position where in reality, the position is negative, but the model predicted it as positive. So these are your false positives. And in the bottom left-hand corner, you have where the position was active, but the model predicted it as inactive. And so these are your false negatives. You'd like to minimize the percentages here. And so when we look at a dummy classifier, we can see that basically everything is sort of equally distributed. And this is what you would expect for a model that's not providing any actual information. If we move over to the right, I'm showing now for 3 different variables that were recorded experimentally, being the soluble yield, the total yield, and the soluble fraction of the positions that were mutated to acridone. We now have my machine learning models. In the first case, I actually used a Bernoulli naive Bayes. In the total yield case, I used a kernel ridge. And in the soluble fraction, I used a support vector classifier. As we can see, we have small false positives and false negatives as opposed to our true positives and true negatives in each of the cases for these 3 different dependent variables. And this was really encouraging. As we go from the first row to the bottom row, each confusion matrix actually represents the exact same thing. However, in the top row, this corresponds to the model training portion. And then in the bottom, it corresponds to the model prediction on a testing set of never before seen data before. This gives you an ability to sort of gauge how well your model will generalize to new systems. And as we can see, the models perform well in both the training and in the testing. And this is exactly what we'd like to see. Finally, on the bottom right here, I'm showing just a little bit of a what's called feature importance analysis that allows us to see, okay, well, the features that we were able to calculate from biomolecular simulation were effective in making predictions of tolerability to acridone incorporation, but which features were the ones that were important? This analysis allows us to see that. By doing this, we were able to observe that terms corresponding to van der Waals attraction and repulsion were important, as well as structural terms corresponding to things like the omega angle, the phi psi angles of the residue, as well as the side chain rotamerics, and then things like the different ways that we can compute the implicit solvation energies. And this should make sense given the structure of acridone being a large hydrophobic amino acid. With all of this, I invite you to check out the rest of our paper and reach out if you have any questions.